This is a celebration of the completion of 1 year of presentations here in seminar at the Controlled Environment Agricultural Center in the College of Agriculture and Life Sciences in Tucson, Arizona, the University of Arizona. And I'm Gene Giacomelli, and it's a pleasure just to say a couple of words about where we've been this year. We started out with mushrooms, fungus, studying fungus, Dr. Barry Pryor. We moved down to controlled environment, organic vertical farming with Ed Horton. Went to Mars with the two engineers from the Jet Propulsion Laboratory, and then studied a bit about the nutritional concerns of food wastes and what we could do with them. Moved back into vertical farming, actually a couple of times. Brought in Merle Jensen to discuss about the revolution or the evolution, now he likes the word revolution, of controlled environment agriculture and how we were all a part of that, and as we speak, we're continuing. Another vertical farming discussion with Mark Ferguson of Hungry Planets. Dragos was here talking about cracking the code of soil genesis, and he's got that all figured out. And then John Adams from Biosphere 2 came last time to speak about Biosphere 2 involved with Biosphere 1 and beyond. Today, we wrap up this year with another faculty from the University of Arizona, Rod Wing, who will be introduced shortly by Rafi. But those who had the opportunity to see the cake, we will enjoy it while the man is speaking. Those out there, I'm sorry, we cannot send you any samples at all, but we encourage you to come back next year when we'll have another cake, I hope. Anyway, Rafi, are you ready for an introduction? Thank you very much. We look forward to a great presentation.
So again, thank you all very much for sticking with the program and coming to hear the last speaker of the academic year. And before I introduce Rod Wing, I have a couple of questions for you. The first one is, how many of you know what 10BPQ is? How many of you? Just raise your hand. You didn't read my... that says something. Okay. The second question along the same lines, did you notice anything strange or erroneous on the cake? The word environment has been misspelled. But you didn't pay attention to that either. Okay, enough with the levities. I'm really thrilled to introduce today's speaker, Rod Wing, who has more titles than I can fit in 5 minutes. So I'm not going to name them all, but I wanted to highlight for you the 10BPQ that he's going to be talking about. And since only 2 of you raised your hands, 10BPQ means the 10 billion people question. And the 10 billion people question is something that's going to come up in 2050, according to the best estimates, right? More or less. And the idea is, how do you feed 10 billion people on this earthly planet that we all inhabit? And Rod Wing is in the leadership position of moving very rapidly toward the elucidation of, first of all, the genomics of the plants that provide the majority of proteins to the world today, and the quest for developing new plants of the same kind of species that grow better, produce more, require less water, are resistant to disease, and grow in less land. It's a fantastically difficult question to solve, but it has to be done, and he is a man who is already doing it. And so I don't know if you noticed, I labeled all the foods that were prepared for today with the scientific name. I thought it was important for all of us to recognize that the food that we eat actually comes from plants. And the elucidation of the genomics of plants is here to tell us all about it. Rod Wing, thank you so much for coming and sharing your knowledge with us.
All right. Is my mic working? Okay, great. Thank you all for thanks for the kind invitation, the excellent introduction, and it's really exciting to be here. I always like coming up here. It's great to be around plants. I'm not around plants enough. I'm around a bunch of physicians and engineers, and not around plant folks quite enough. So today, maybe we could turn the lights down a little bit, or is that going to affect anything? Okay, I kind of like to have them down a teeny bit. So, okay, let's hope this thing works. Trying to get okay. Sorry, a little technical. Let's see if it's going to continue to work. Okay, good, good. Okay, so I'm going to talk about natural variation across the Oryza genus. That is the genus that I've been working on for many years, and this is the genus that contains the two cultivated species of plants, Asian rice and African rice. So today is my outline. We're going to talk about, first I'm going to have a little introduction to the Arizona Genomics Institute, which I direct, and then we'll talk about natural variation in crop plants, in rice cultivars, as well as natural variation in the wild relatives of rice. Then I'm going to discuss a project that I've been running since 2003 called the International Oryza Map Alignment Project. And then we'll talk about one of the genomes that we recently sequenced, the genome of African rice. It's the story of black rice. It's a fascinating story. I'll wrap this up with a discussion of a recent position that I got, the AXA chair in genome biology and evolutionary genomics at the International Rice Research Institute, which is permitting me to translate a lot of the genome biology that I've been working on for many years into practical solutions to help solve world hunger. Okay, I'm going to start out first introducing my group. I direct the Arizona Genomics Institute. We've been here since 2002. We have several groups that are part of the unit. We have a functional and evolutionary analysis group. We have a sequencing group, bioinformatics group. Bioinformatics is the computational understanding of genomic information. We have what's called a back library construction group and a physical mapping group. I'm not going to get too much into that, but this is really how we got started in the business of genomics, and that has allowed us to play major roles in many sequencing projects. Currently, we have three group leaders: Dario Capetti, Dave Kudrna, and Jean-Louis Zhang. Sequencing, kind of more institute management, and then in bioinformatics. And this just shows we're located in the Keating Bioresearch Building, north of Speedway. This is more on the medical school campus, and these are just some screenshots of some of the things we've been working on over the years. Our group has played a significant role since coming here in 2002 in over 30 genome projects. The first one that we were involved in was the draft genome of rice back in 2002, and then we actually created, we led the U.S. effort to sequence the rice genome to generate a kind of a reference standard that still today is the highest quality crop genome of any plant. We worked on the maize genome, the cacao genome, soybean, tomato, the 12 genomes of Drosophila, an experimental fly system, and our most recent genome was published in September 2014, the genome of the African rice, which I'll talk to you about later. The big question that drives us and that I think drives most people in agriculture is really what we call the 10 billion people question. And that is, again, how are we going to grow enough food to feed an additional 3 billion people on the planet in less than 40 years? It's a serious problem, and we all have to be involved in trying to solve that solution. So rice is going to play a huge role in that because rice feeds about half the world already, and it's the rice-dependent population that will double by 2050. So what the rice breeders are working on is trying the development of new varieties of rice that are high yielding but more sustainable, ones that require less water, fertilizer, pesticides, have reduced greenhouse gas emissions. Rice is a leading producer of methane emissions in the world. And we call these crops next-gen super crops, or green super rice is another name for these. So before I talk about that, I just want to talk a little bit about rice production in the world. So are there any ideas on who produces the most rice? Is the question? India? India and China, okay? They produce about half the rice in the world, and these are essentially the top 12. Brazil is the number 9, and so in the New World, they're the largest producer in the New World. And then here comes the U.S. At about 1.2% of rice. And I'd like to let you know that within rice, well, in the United States, the majority of rice is grown along the Mississippi, the Delta, Arkansas, Texas, Louisiana, Missouri, and also about 40% of rice is also grown in Northern California, in the Sacramento Delta. But we are in Arizona, my lab is the largest producer of rice in the state of Arizona. And this is right here in our greenhouses on the 6th floor of the 6th Street garage, on the top of the roof of the 6th Street garage. It's a good place for growing rice. There's just not enough water here to grow rice in Arizona. But rice is a good model system for studying other cereals, and it has the smallest genome of the cereal crop. So that's why we really worked on it, and it has a lot of shares a lot of evolutionary history with wheat and maize. So in 2004, we announced the completion of the rice genome here in Tucson, and we had a 10-year anniversary last year, and now, but when you have a genome, that is really just the beginning of the project. You think that you have all this big effort. It's a $200 million effort to sequence the rice genome, and once you finish it, you think, okay, this is it. We've accomplished something, but it's really the beginning of a huge project. So what we're interested in is trying to discover natural variation in rice, and you can essentially find diversity in rice for almost any conceivable trait. So this is just a slide showing various seeds shape, seed size, seed color, panicle architecture, plant architecture, and all this variation exists in gene banks around the world. So we, in particular, work with the International Rice Research Institute in Los Baños, the Philippines. That's where miracle rice was developed 50 years ago. That helped was part of the first green revolution. And Erie has 125,000 rice accessions. And recently they sequenced 3,000 accessions, resequenced 3,000 accessions, which means they took 3,000 diverse accessions, grew them up, isolated the DNA, sequenced them a little bit, and mapped all that information to this rice reference genome to look at differences between each accession and the reference genome. And they came up with what's called, this is an admixture graph. And these 3,000 accessions can be divided into 15 different subgroups. These are the Japonica groups. The Japonica is more of the sticky rice. These are 8 indica groups, and there's 2 aus, which are very drought tolerant, and there's also an aromatic group in here, and this is called the admixture. So if you could take these 3,000 accessions, you can actually divide these into 15 different distinct groups, okay? Now, and they all have a tremendous amount of variation that what we want to do is be able to try to capture that information to breed new varieties that are going to feed this the green super rice, the next round of green revolution. So how are we going to do that? So this is a photograph of the farm at Erie, the International Rice Research Institute, and folks who developed these accessions, the sequences for these 3,000 accessions, now plan to grow these 3,000 accessions in upland environments as well as irrigated environments, and phenotype these accessions under varying conditions. And we're calling this the global rice array. We want to do this in the Philippines and many locations around the world, both in the field and in the greenhouse. And so how are we going to do that? In the field, there are these tractors now that have booms on them. They're called phenotyping tractors. This is the high throughput phenotyping field scanner developed by Erie. What you have are 25 plants in a plot, and these scanners go over the field, and you can do 3,000 plots an hour, and you can take a number of different types of measurements. And you associate like the biomass, canopy cover, plant height, stresses. You map those onto, you phenotype those onto these plots, and then you already have the sequence of that plot, so you can associate the phenotype with the actual genomic location of that particular trait. So these are GPS auto steer tractors. Another way of doing this type of phenotyping is gantries. Here's a large gantry that was recently installed in Maricopa at the Maricopa Ag Center. This is part of a huge project, bioenergy project, to look at grain sorghum. I don't know if anybody has seen this yet. I was just in Maricopa on Monday, and we're talking with the folks there at MAC and ALARC to try to plant this rice array, the global rice array, in this phenotyping unit, and from seedling to full seed. Another way of doing genotyping, of high throughput genotyping, is in the greenhouse. And let me see if I'm going to make this thing work here. No, sorry. Okay. Now, what you're looking at here is a phenotyping greenhouse in the Research Triangle Park. This is owned by Monsanto, and what you can do is essentially plant your array, like this global rice array, and the plants are grown under extremely controlled environmental conditions, and you can do numerous measurements, and again under controlled conditions. So we can define and we can map these traits, and then again we'll have a list of traits, and we can say we want a plant that flowers at this time, we want a plant that's this height, this many tillers, this type of seed, this type of resistance, and you can use that information to accelerate the breeding process. The reason why I'm showing this is I recently gave a seminar in the Research Triangle Park, and this Monsanto facility is actually closing down. And I was telling my colleagues at this, one second, let me get this. So what do I do here? I do this and this. Okay. Click, what do I do now? Okay. Let's see, that's good. Yeah, we're good. Okay. The reason why I wanted to show this specific facility is because the Monsanto is essentially closing their this facility, but they're not going to pack up the greenhouse. And I talked to my colleagues at University of North Carolina, Duke, and North Carolina State about the possibility of trying to take this facility over and plant the global rice array in there and do significant amount of phenotyping. And negotiations are underway right now to actually try to take this facility over. And there's actually a possibility that there might be kind of a donation of a facility like this for the greater good to donate something like this to Erie and ICRISAT, which does a lot of work on sorghum, for example. So anyway, this is something that, it's already there. It's already built. It's maybe a $20 million facility. So this might be actually integrated into a kind of a national phenotyping center. But I'd love to have one of these things in Arizona as well, which is what we've tried to do, but I think there's still an opportunity to do this. Okay, so in addition to the looking at natural variation already in cultivated rice, our lab is very interested in understanding the genetic variation in the wild relatives of rice. This is a photograph of some of the wild relatives of the genus Oryza. These are the two cultivated species. This is Oryza sativa, Asian rice, Oryza glaberrima, African rice, and various wild accessions. These are essentially weeds that you would find in the field and under various environments. But they're agronomically inferior, but contain virtually untapped reservoir of genetic variation that could be used for crop improvement. The example here is Oryza cortata. Cortata can grow in salt water. Okay? If you can imagine transferring the genes from cortata into Asian rice, we could maybe irrigate our rice with ocean water or diluted ocean water. And the nice thing about working with the Oryza species is that we can cross all of these wild species with the cultivated species. There is sterility, but you can do you can do back crossing, so you can actually introgress segments from the wild relatives into the cultivated species. This is what's called a phylogenetic tree. This shows the relationship of the various species amongst each other. This represents about 15 million years of evolutionary history. The Oryza has 23 species, wide distribution and habitat, 10 distinct genome types, and a 3.6 genome size variation. The AA genome types are the primary gene pool for cultivated rice, again, sativa and glaberrima and the wild relatives. We have 4 polyploid species, and if you look at the some of the wide distribution and habitats, we have material that grows in Australia and South America, Africa, all throughout Asia, all kind of in the tropical regions. So you can find these wild relatives in many regions around the world. Africa is one example. Africa has the most diverse amount of genome types. It has 3 AA genomes, a BB, CC, and an FF. Here we have African rice here, the progenitor Barthy, and then Longisteminata. Longisteminata was the first wild species where the first disease resistance gene was cloned out of plants, XA21 by Pam Rollins' group at UC Davis. This is their range in Africa. The African species have a treasure trove of traits. This just shows some examples of traits associated with biotic and abiotic and biotic stress, drought, high mineral content, and then bacterial, viral, fungal, and insect resistances have all been found in these species and have actually been crossed into cultivated rice already. So my lab started a project once we sequenced the rice genome called the Oryza Map Alignment Project, and that's now morphed into the Oryza Genome Evolution Project. And the goal was to essentially create what we call a genome-level comparative system that can be used for both basic and applied research. So this is a voucher plot, and this shows a number of the varieties in the greenhouse at Erie. And these are the investigators involved in this project. Initially it was Scott Jackson, Lincoln Stein, and Doreen Ware in OMAP, and Doreen and Scott have stayed on the project. And then we integrated 3 card-carrying evolutionary biologists onto the team. Mike Sanderson from Arizona, Carlos Machado, who works on population genomics, and then Manuan Long from Chicago, who works on the evolution of new genes. So it's with the groups. Our work has been going on since 2003, and we generated at the very beginning. We generated lots and lots of resources for the project. We generated libraries, physical maps, chromosome 3 short arm sequences, 3 reference genomes, glaberrima, Barthy, punctata, and Liercia, which is an outgroup, a lot of RNA data, and a lot of resequencing of populations of the AA genome species. Done a tremendous amount of analysis, publications, and the majority of the data can be found at grameen.org or omap.org. And what we've come to the conclusion by analyzing all this data is that a single reference genome that represents the entire genus Oryza is essentially insufficient to capture the allelic diversity that is trapped within these wild relatives. So what my group has done is we organized a consortium of scientists around the world. A lot of these people were involved in the original rice genome sequencing project. We call it the International Oryza Map Alignment Project, and we've held grand challenge meetings since 2007, and our goals have been to generate high-quality reference genomes for representatives of all of the species of rice, including all of the AA genome species, generate mapping populations that can be used for breeding studies, for functional analysis, and identify naturally occurring collections of these species for diversity, conservation, and population evolutionary analysis. So kind of to preserve these species for future generations. So today I'm going to talk primarily about the reference project. And what I'd just like to say is that we now have, of the 23, we have 14 genomes that have been completely sequenced, and they're considered reference genomes. These are the people that have been involved in it. I won't go through this list, but we have genomes now for all the AA genome species, BB and FF, Brachyantha, and then the 3 CC genomes, and what's called an outgroup. And the 2 remaining diploid species are Granulata and Australiensis, and these are the folks that are involved in this project. So, what I'll talk to you about today is an analysis of 11 of those genomes. We call this the 11 genome Oryza plus Liercia data package. These are the genomes, and what we've done with these genomes is we've analyzed these sequences for what are called transposable elements. We've looked at their gene expression, we've looked at their methylation, and then we've put all this data into what's called an annotation platform, which takes the genomes and then it finds all the genes and finds where all the transposable elements are. So we have to essentially make a map. So we have the genome. Now we want to know where the genes are. Once we know where they are, then we can compare and contrast the various genomes. That's kind of what we're trying to do. And this is a paper that was recently published by Dario Capetti and John Wei-Shang in my group, describing a database for repeats. So repeats, it's very important. Genomes are scattered with repetitive sequences, and you need to define these repetitive sequences so you can find the genes from the repeats. So this is something that has recently been done. And then all this data, again, is run through Maker, which is an annotation tool. And what it does is it takes all this evidence, and then it gives you kind of the structure of a gene. And this is a prediction. So all this work has been done, type of work has been done for the human genome, for the fly genomes, for rice genome, and now for the Oryza genus. This shows the number of gene models, and it's very similar across the 12 chromosomes for all these species. So this looks good, and this is the final kind of data. These are the size of the genomes, the number of repeats. So the repeats, actually almost half the genome is composed of repeats. The number of loci and the number of, I'll just stop there. So what have we found? What have we discovered with all this data? So now we have the genomes. We've now annotated them. So what are some of the highlights? So I'll tell you a little bit about that. First of all, we've been able to time some of the speciation events. And if we look at the speciation of all the AA genomes, it looks like that occurred about 2.5 million years ago. So we essentially have a new species every 500,000 years, which is pretty, this is something that you would expect on island radiations, but you don't really see this on continental radiation. So this is, we say that the diversification in Oryza has evidently gone hand in hand with circumtropical dispersal rather than extreme local adaptation to different environments observed in island radiations. So that's kind of a new highlight. Another highlight is this is called transposable element turnover rates. We've been able to measure turnover rates now in the AA genomes, and it's one transposable element every almost 2 million years, and that's 15 times to 800 times higher than you see in flies and mammals, which is pretty remarkable. That means the genomes are turning over, they're stable, but the transposable elements are turning over very, very rapidly. We've also done a number of comparative analyses of a number of traits. One is there's a trait called submergence tolerance. This is the sub-1 genes. We've looked at those. The sub-1 genes allow rice to be flooded for up to 2 weeks and still survive. And we discovered a new sub-1 gene in the wild relative, Oryza pierii, the outgroup. We've looked at phosphate utilization, shattering, and disease resistance. So I'll tell you a little bit about disease resistance genes. This is a complicated slide, but I just wanted to say that now that we have these 11 genomes, we now have a whole collection of putative disease resistance genes that can be used for crop improvement. And so the idea is now we want to discover these genes, but we also then want to figure out a functional assay for these genes. So one thing that Josh Stein looked at was, first of all, we found about 4,500 genes, disease-resistant genes, across these 11 genomes. And one thing that he noticed, Josh noticed, was that there's this type of disease resistance it's a hypothesis called a decoy model, where they've identified heterologous disease-resistant genes that are oriented head-to-head. So they share a common promoter, which this promoter regulates gene regulation, and then you have one gene that's a decoy, one gene that's the disease resistance gene. And so he's identified numerous instances of this phenomenon in rice, and this just shows an example here of one set of our genes that we have 4 out of 5 are in a head-to-head configuration in Oryza sativa japonica, and then 2 out of 2, and then all the way down to 2 out of 6, all the way in Leersia. So over 15 million years, we have this conservation of head-to-head configuration of resistance genes. And this, we feel that this is significant, and now we need to figure out work with plant pathologists to test our hypothesis to see if this configuration actually is going to allow us to identify genes that are going to be important for disease resistance in plants. Okay, so that's kind of a highlight and of some of the analysis. And so now I want to talk a little bit about the African rice genome, or we also call this black rice. This is our most recent genome that we published in 2014. The reason why I show this slide like this is we were motivated by this book by an author from UCLA. Her name is Judith Carney. Her book is essentially about the impact of rice cultivation on early American history. And there was a time in the 1700s where South Carolina was one of the largest exporters of rice in the world. How can that be? The slave owners would say, we taught the slaves how to grow rice. But in reality, the slaves all came from rice-growing regions in Africa that were in South Carolina. There was right before the Civil War, there were 1,600 plantations and farms that were growing rice primarily in South Carolina. They were producing 182 million tons of rice that was exported. And 2/3 of that was from South Carolina. So that's about 90% of the rice was exported. Another 10% was used, to feed for subsistence feeding. So her hypothesis was, her book is about how she built the case where they provided evidence to support the idea that slavery was not just about muscle, it was also about the transfer of knowledge from one culture to another. So we tried to tell that story in the genome. And last year in the Tucson Book Festival, Judith told her story of black rice, and I told our story of the rice genome. So we sequenced the genome of CG14 of Oryza glaberrima. This is the parent of a rice in Africa called Nurika. Nurika is new rice for Africa. What that is it's a cross between African rice and Asian rice, and then back crossed many times to Asian rice. And so Nurika has the high quality, high yielding properties of Asian rice and the adaptive properties of African rice. And Monty Jones got the World Food Prize for developing these Nurika lines. So we did, we sequenced the genome. We generated gene expression data. We did annotation, and then we did baseline annotation, which is just this: find the genes, find the repeats. And then we did topic-specific annotations. We looked at domestication, flowering time, light response, structural variations. So we asked two questions with this data, and a little bit of additional data. So the first one was African rice. So Asian rice was domesticated in China thought to be along the Yangtze River about 10,000 years ago. African rice was domesticated in West Africa 7,000 years later in a completely different species. So 2 independent domestications in 2 different species of rice. So we asked, there are 2 theories that this domestication occurred many times in Africa or in a single domestication center. So what we did to answer that question, so the one of the theories is that rice is domesticated along the Niger River in what is now Mali, and then was diffused to 2 secondary centers along the coast. So Muwa Wong, a graduate student in my lab, did a experiment where we looked at the population structure of the progenitor of African rice. And I'll tell you what that essentially is what we did was we took 94 Oryza barthy accessions that we collected from Africa rice, which is in Benin, West Africa, and we sequenced them, and then we grouped them. We looked at their variations, and then we were able to group these 94 accessions into 5 groups. These 3 here, 5 here, and then what's called an admixture. And then we just simply asked the question, where are these groups from? So you would think that one group of Obarthy from one region that's going to have similar genetic information, and the further they're apart, the more diversity you're going to have. And that's the case. This accession, this group here, and the admixture of Barthy all came from West Africa, of this part of West Africa right here. And as you move further away, these accessions became more predominant. So what this tells us is that our data, looking at the progenitor of African rice, supports the hypothesis that there was a single center of domestication. We then asked the question, did Asian farmers select the same traits and the same genes as African farmers 7,000 years later during the domestication process, or were they completely different? So we did a series of tests, and I won't go into those, but we identified 5 genes that are known to be domestication genes in Asian rice that appear to be selected for in African rice. And these are the 5 here. A shattering gene, semi-dwarf, this is a gene that makes the plant small, a gene involved in panicle architecture, a gene involved in aroma, which was a surprise, and a red pericarp gene. So I have a big interest in abscission, in shattering, from my early work in tomato. So this is what shattering is in rice. Okay, normally the cultivated rice, pretty much normally all the time, this is what a panicle would look like. The seed stays on the flower. The wild relatives, they all shatter, and this is an evolutionary trait for seed dispersal. Okay, and so early farmers, you would predict would have always, they would find naturally occurring mutants that no longer shatter. Okay, so the shattering pathway is pretty well understood in Asian rice. So we looked at the several shattering genes, and I'll show an example of 2 shattering genes in African and Asian rice. So here is the Oryza sativa. This is a region that contains OSH1, a shattering gene, and then there's 5 downstream genes and 2 upstream genes. And there's a single point mutation that essentially makes it so that the gene is no longer expressed. It's a premature stop codon. So this is the causative mutation in Asian rice. Okay, so now we look at Barthy, OB right here. It has essentially the same exact structure as sativa. the 5 upstream, the 2 downstream, and the gene right here. But there's no mutation here, so the Barclay does shatter. Okay. However, in African rice, in glaberrima, we have a 40 kilobase deletion, so this whole region is completely missing, plus this region is inverted. Okay? So, so what this told us was that this gene was, the same gene was selected for, but the mutation that was selected for was completely different. Here, the Asian rice uses a point mutation here. The African mutation is selected for a 40 kb deletion. This is another gene, shattering 4. This is the causative mutation in Asian rice, and everything else is what's different between Asian rice and African rice. You have a number of point mutations, 3 deletions, and 2 insertions. So combined, we came to the conclusion that, at least in the shattering pathway, that early African farmers selected for the same traits, the same genes, but the mutations were completely different. And we had to prove that because Asian rice and African rice now grow together in the same fields or nearby fields, and you could imagine that the domestication genes from Asian rice would have been crossed, could have been introgressed into African rice, but that is not the case in our study. Okay, so I'll just kind of wrap up with this little section right here and just say that we have this 11 genome data package, and what I've been working on for the past, I'm going to say 6 months, is putting 19 manuscripts together that have been recently submitted to Nature. We call this the Oryza Nature package. So these manuscripts describing a lot of this work are now under review, and hopefully we'll have a big splash in November on all this. If you want to see the data, you can go to gramine.org. So the next genomes that I'm interested in sequencing are the polyploids. And our first one that we're going to go after is cortata, the salt-loving beast. So how are we going to do that? I'll do a couple more minutes here, and then we'll wrap it up. So I recently was awarded this AXA chair for genome biology, and this is again to try to work on translating some of the genome biology that I've been doing into functional work at Erie. And one of the things that's needed really is we now have 2 or 3 reference genomes that would represent that, but we still do not have enough reference genomes to represent all of these 15 subgroups. So we need this as a tool so we can discover all of the allelic diversity present in these genomes. So our philosophy at the Arizona Genomics Institute is that the first genome of any species or subpopulation should be as high a quality as possible. And most of the genomes that people work with these days are actually pretty crummy. So we've been focusing on really high quality genomes. So our lab was fortunate enough to be able to purchase what's called a PacBio RS2 instrument. What this machine will do is give you sequences that are on average of almost 20 kilobases in length, where we're used to sequences that are only maybe 200 bases in length. So it's a huge advance. And we've recently sequenced the genome of N22, which is what it's called an Aus rice variety, and this is a highly drought-tolerant variety. And we've been able to get some very good data out of this genome. So we're having, this is called a Contig N50, but what that means is our average sequence lengths after things have been assembled are 906,000 bases long, so they're huge. And this data is being released shortly. I'm going to just wrap things up and just say thanks a lot for having me speak. And this rice project is really a global initiative, and we're working very hard to develop resources that can be used by the entire world to help solve this 9 billion people question. And again, our strategy is to try to look at natural variation. I mean, genetically modified organisms are very useful, but that's really kind of one gene at a time. There's so much variation that already exists in these crops, the wild species, as well as all of the accessions that are in these gene banks, that what we want to do is capture that and be able to design new crops as quickly as possible to really help solve this problem. So this is all my collaborators, my team of kind of now and previous staff, and then we've been funded by the National Science Foundation, the Tannemer and Antle Endowed Chair, and the AXA Research Fund. And with that, I'll show you my view of the world. Thank you very much.
Thank you, Rod.
You're welcome.
How did that go? Let's see. Yes, applause. I don't think they can hear that on the internet.
Nobody ate the cake.
Nobody ate the cake. We didn't get to it.
Yeah,
We're
yeah.
just enamored with this analysis here. There must be a number of questions. All right. I hope you're good.
Yes.
My name is Dragos. Now, there are, as far as I know, there are more than 9,000 varieties of rice only in India, right? Am I correct? How much effort is put into understanding those traits that, having so many varieties, you would hope that they are adapted to a wide variety of climatic conditions, particularly, with the climate change now affecting the Earth environment. So how much those varieties are studied?
Within the 3K set, there's a number from India, for sure, absolutely. Everything that's in this in the Erie Gene Bank is essentially what has been donated to the gene bank. So right now, the kind of walls are building up to now to get new genetic material out of India, to get it out of China. It's really hard to get this material out now. But just rules and regulations. It's like that you might be getting something that's has intellectual property, and I think that's short-sighted, because if all these barriers go up, then we're not going to be able to exchange material freely, and we might find a new allele in an Indian line that is going to be extremely useful. So really, the long-term goal is to now go in and kind of resequence the entire gene bank collections. The 3,000, I mean, that's not really that much, and we're still missing tons and tons of rare alleles that could be useful for crop improvement. So the Indians are definitely involved in a lot of these analyses. So so it's really, and China is, I would say more active. Thanks for your question.
I have two questions. You mentioned miracle rice. Miracle rice. Could you explain that a bit?
Yeah, this is developed in the 70s. It's a well, actually, its 50th year anniversary is this year. Okay, so when people were growing rice years ago, rice was generally very tall, and it would undergo lodging if typhoons would come along. So what happened? So what miracle rice is, essentially, it's like a high-yielding Indian variety called Pita, and it was crossed with variety from Taiwan, Doji Gowa, which is a very little short guy. All right, so they put them together and then did a series of back crosses and developed what's called IR8, and that was miracle rice. It was extremely high yielding and lodging resistant. But they had a problem in that the grain was very chalky. So people could grow this food, but they could eat it, but it tasted terrible. So now there's IR64 is really a very popular variety, but then things have gone way beyond that. But that's called miracle rice, and it's part of the green revolution of rice.
Thank you. My other question is, so there seems to be a lot of current trending towards pushing towards. I forgive if this is inappropriate, but like GMO banning and whatnot. You're doing natural selection type stuff. Could you just speak to that a bit? How that's going to affect the landscape of future crops and whatnot?
Yeah. So everybody, whenever I give a talk about plants, everybody asks about GMOs. And I always have to defend it. I mean, I think GMOs are fine. But,
well, me too, but a lot of countries are pushing towards banning GMO.
Yeah. But, you know, my view is, though, that I think there's a place for GMOs, but really there's so much natural variation out there that we need to focus a lot of effort on that, because this is what evolution has done. I mean, evolution has, I mean, the genes that didn't work, they're gone, right? So now, evolution is out there, and this is all captured. So we want to try to capture this, and you know, you find, let's say you find a combination of genes that work really well, but you want to tweak that a little bit. You can go into the natural variation collections and find the tweaks that have already been made and make combinations really quickly, and you don't have to worry about what's called linkage drag, where by breeding in something that's very, very distantly related and having linkage blocks that won't recombine. So, but you know, for banning GMOs, I think that's a bad idea. I think we need everything I mean, this is a huge problem. So, I mean, people will. I mean, my example I say is there's everybody. There's so many diabetics out there, and they're injecting insulin into them. That's a product of recombinant DNA, which is the same thing as GMO products. It's a product of recombinant DNA, and you know, you can inject a drug in you, but you can't eat a GMO corn. So I don't know.
Yeah, because I think if I'm following right, you can use natural variation to find the genes, but then as far as, say, like creating a certain variety, it would be a lot of work to naturally breed that out rather than GMOizing, just programming in the genes you found naturally occur.
So what I'm promoting is more doing it through conventional breeding, because we have I mean, we have markers now for these genes. If we want to bring in an allele that we think is going to be very good, we already have a marker for that. So we can do the cross and track that through an advanced breeding program. In rice, you can get maybe 4 seasons a year if you really push it. So you can accelerate the breeding if you have the right molecular tools. So, and there's also that CRISPR technology that's out, which they're still trying to decide if that is GMO or not. Yes.
Hi. Beyond using natural variations versus GMOs, what would you say differentiates the way we're going about solving this big question versus others out there?
Well, it's you mean solving the 9 billion people
right, the
question.
9 billion people
right,
question.
Right. So we're trying to our approach is to at least just try to develop new varieties that are higher yielding and more sustainable. Okay, ones that, again, that will require less water, fertilizer, pesticides, and can grow on marginal lands and have reduced greenhouse gas emissions. I mean, this is no matter if this this approach is going to give us crops for the future and to protect the environment in the future. So that's a worthy goal, and this is something that we need to do for all crops. Okay, but there's lots of other. It's not just crops, right? I mean, people would say we already have enough food, we're already growing enough food, but it's just really there's a lot of waste, right? And the last time I was in India a couple of years ago, there were farm subsidies. They were saying, okay, we already know what the price of rice is going to be. It's going to give us a top dollar, so they just grow it, and the government buys it, but they don't have any place to put it. So it just sits there and rots, you know, in the fields and stuff like that. So it's crazy. So just simple things like building grain elevators or ways to store rice long term is there's a lot of simple things. I was told at Erie that laser leveling of fields. Most fields are going this way, that way, right? So, and the yield loss is amazing. If you can laser level a field and that'll last for maybe 5 years, your yield goes up 20%. Just simple, really simple. That's an ag engineering problem. So it's not, I'm on the molecular biology breeding side and things like that, and I think it's going to take a combination of many, many things, and political barriers of like sharing germplasm. That's a big issue right now. I mean, it's because people they think, okay, if we give you this line, it has that magic gene in it that's going to be worth millions and millions of dollars, so we're not going to give it to you. And it's crazy. We need to work together as a team to make this happen.
I had one more question. Nutritionally, is there a difference between the varieties of rice that we're looking at? And does it make sense to focus on one that might be more nutritious versus another?
There are differences in nutrition for sure. Some have higher protein. Some have. I mean, like the vitamin content. I mean, the one thing about rice is they're actually the vitamins and oils are actually there in the rice, but we take it all away when we polish it. And the reason why we do that is because the hulls have oils in them that for long-term storage, if you don't store it right, it'll rot. Okay, so it goes rancid. So that's why we throw it all away. If everybody was eating brown rice, that if you grow it and you store it properly, then that solves a lot of problems nutritionally that way. But not everybody wants to eat brown rice. I mean, it's a very, like you said, there's 9,000 varieties in India, right? I mean, it's like every little region, every little town has their own rice that they'll eat, and it has to be this way, it has to be this color, it has to have this smell. So, but I think like improving protein content is important, vitamin content, cooking quality. There's all sorts of things out there that people are working on. Yes.
Rod, thank you. This is Jerry McCarty.
Yes. Hi.
Hi. In the face of the drought that we're the nation worldwide facing currently, and tied into an up and down factor on that, but the availability of water, and at one point you had mentioned salinated water or ocean water as a alternative for the hydrology factor. And can you elaborate a little bit more on if there's been more of a long-term study on the rice with salt water?
Well, so I mean, there are more salt-tolerant varieties because, as you cultivate land, the salt content actually increases over time. All right, so the crops that you put in now are not as good as are not as resilient as ones that the same crop would not be as resilient because it's having to fight that salt. So you have to get salt-tolerant lines. Okay, so that is ongoing. Crossing the cortada into sativa, that has only recently occurred a couple of years ago. So they're still trying to work out this crossing factor. And the thing is that cortada is so distant that when you cross in salt-loving genes, you're also bringing in 1,000 other genes that are detrimental. So you could get a plant that loves salt, but the yield is just terrible, and the quality is just terrible. So it's kind of sorting that out. So this is where technology of GMO or like a CRISPR thing would work out. But still, that whole we haven't figured out the genes that are involved in the salt tolerance yet, and so that's why at Arizona we're going to sequence the cortada genome to figure this out. Yes.
Thanks for the presentation. So in one of your slides, you mentioned that there's going to be use less water and less pesticides. And just looking at the GMO right now, we have the Roundup resistance. That type of pesticides actually toxicity-wise is much more than some of the other type of pesticides combined together. So for the new species that using less pesticides, how do you guys determine and justify the toxicity of this new type of pesticides you're going to use?
Well, I guess what I'm saying there is that because now we have, I showed you that phylogenetic tree, so now we have kind of 15 million years of evolutionary history at our reach, and so we can pull out resistance genes and identify resistance genes that are going to allow us to maybe be more long-term resistance genes for insects. I mean, I would rather use a disease resistance gene for an insect than spray the field with a pesticide. That's what I want to do. So this allows us this opens the door for the discovery of a whole new array of genes that we can test, and some of these are going to be durable. So I mean, so Roundup Ready. I mean, Roundup that you're thinking that's more of a weed killer. So that's more you kill the weeds and you don't kill the plant. So that's what that's primarily for. And I think that's a great idea, and it's biodegradable, and it when I would like the Bt toxin, that's a naturally occurring gene in a bacteria that they put into a plant. Okay, and so like when I first taught at Texas A&M, the cotton fields, they would spray the cotton fields with insecticides every 2 weeks, and whereas now with the Bt resistant cotton, it's maybe twice a season. So to me, that's a lot more environmentally friendly than having to spray all the time. I'd rather live next to a GMO field than a non-GMO field, personally. I don't want to get gassed to death.
Okay, one more question.
Losing my voice, I think.
With the water scarcity worldwide going, and water being the rice being the wetland and water-loving plant, and also in the hilly region, and there is leveling the field is a kind of a problem. Either just wonder how much emphasis is being given to develop or in upland rice.
No, I mean, so in this panel of 3,000 accessions, we have there's a number of upland. I think about 500 of the 3,000 are upland varieties. So I mean, there's definitely a push for that. And so this field in Maricopa that we're going to try to plant the 3K set, that first batch that we want to put in is more of an upland variety, a set of upland varieties to do phenotyping with. So I mean, you got to work with both. The thing, with upland, that's going to conserve water, but they're not as high yielding, and you really only do like maybe one crop a year for that. So.
Okay, lots of trade-offs, right?
Yeah.
We have to deal with a lot of different a lot of different things, even agricultural engineering and land level, right? So they get 20%.
Do you guys teach that here?
No, we gave
Laser
up on
level?
that a long time ago, but that's because it's out there in the real world and people have it figured out.
Yeah,
so they can
you don't
do
have
that.
to deal with that.
No, we don't have to deal with that. Rod, thank you very much, and let's give him a applause. And I want to thank all of you for coming, and I'd like to thank all those online for being here. This is the last seminar for the semester. You can go to our website and view all of these presentations well into the future. So we've got you on tape, as they say, and we'll go from there. Everyone, I want to thank certainly Rafi for all the work he did this year to make this happen. And yes, thank you. Applause, applause. And we invite you back again to continue with the long list of people we'd like to bring in. So let's do that. In the meantime, why don't we enjoy a piece of cake and look forward to a good weekend. Thank you very much. Bye-bye.